So I would like to share with you some of evidence that there is a link between obesity, AF, and also fat tissue. The relation between obesity and atrial fibrillation is now well established by a few studies, large cohort studies, which show that with year, both in men and in women, if you have an overweight, the risk of having AF is increasing. But probably the most strong evidence and the most striking study have been provided by this group in Adelaide from Prash Saunders. They randomized a group of patients controlled for atrial fibrillation with a BMI over 27, and one group was submitted to strict weight management plus cardiometabolics and factor control, while the other group was only under metabolic control but not weight management. And in the follow-up, they observed that the group of patients for which there was weight management and with a significant decrease in the waist circumference of BMI, it is associated with a reduction both of the burden of AF and also the severity of atrial fibrillation, but also of the outcome of atrial fibrillation. So this clearly indicates that there is not only a sort of comorbidity shared by obesity and AF, but probably there is some sort of link between the two diseases. And so the question is, what are the underlying mechanisms which link these two disease conditions? And still this group in Adelaide reproduced obesity in the large animals, and after a few months of obesity, they perform an electrophysiological study, and they observe that in the atria of this sheep animal, that when there is obesity, there is a number of area with low voltage or silently electrical area in this obese atria of sheep animals. And when they do the histological examination of this left atria, they observe that matching the low voltage area, there is a lot of fat tissue that accumulates in this area. And this fat tissue, which is of course observed at the surface of the myocardium, is also observed in the atria, and we already showed this morning this histology of infiltration of fat tissue inside the myocardium. So this adipose tissue, or also called epicardial adipose tissue, is in fact pretty well known, for instance, in the context of ischemic disease. And what has been shown is that this fat tissue is intimately associated with the myocardium. There is no barrier, such that there is a freely diffusion of factors, both from the fat to the myocardium, but also the myocardium to the fat tissue. And what is also known in other field is that this adipose tissue is a very active biological tissue which can secrete a lot of adipokines depending on the clinical history of the status of the patients. So in the first study that I will not detail here, we show that when you take this secretum from the fat tissue on the human atria, and when you apply this secretum to an atrial myocardium in vitro, you induce a massive fibrosis in a few days of the neighboring myocardium. And just to follow this, I just want to show you that the composition of this secretum, and we compare the secretum from the epicardial adipose tissue and the subcutaneous adipose tissue from the same patients, and we found that the composition of these two secretums are markedly different, and we could identify some candidate factors for explaining the profibrotic effect for the secretum, and notably activin A, which is a member of the TGF family, and by the way, which is also enhanced during heart failure or diabetes patients, and which could be a good candidate. In fact, when we suppress this activin A by an antibody, we suppress the profibrotic effect on the atria. Then we follow on the study of the histology of this fat and the myocardium, and we look at the right and left atrial myocardium. And we found that in addition to epicardial accumulation of fat, there is also in most of the tissue fat infiltration, which is beneath the epicardial layer. And when we collect more samples, we observe that there is in fact two types of histology. One which is the epicardial layer and some fat with very little fibrosis. And there is another group of histology where it's looked like this fat infiltration is now replaced by fibrosis. So we have access to this human material and try to make some statistical analysis, and it's turned out that the group of patients in permanent AF have more of this fibro-fatty infiltration, but clearly there is other patients with not AF which also have this histological remodeling. So we collaborate with a group of Rick Willems and Peter Armer in Leuven. They developed a sheep model of permanent AF using a pacemaker inserted in the left atrium. And we follow this histology, and again we found that with permanent AF, first there is an increased accumulation of epicardial adipose tissue, but there is a transformation of this fat from sorts of regular healthy fat infiltration to this fibrotic and fibrotic replacement of fat tissue in the atrium. So both in this sheep model and in human, we want to understand what makes the transition from this fat to fibrosis. And again, in other disciplines, it is well known that this visceral adipose tissue is a strong factor of fibrosis, and notably through immune response, inflammatory and immune reaction. And we were interested to observe that there is a lot of infiltration at the border between the fat, the myocardium, and the fibrosis of these mononuclear cells. And we characterize these cells. They are, in fact, T lymphocyte, T killer lymphocyte, CD4 lymphocyte, as observed in other tissue. And actually, we are characterizing this immune response, which probably contributes slowly in the process of fat accumulation to its slow transition to fibrosis, and also raise the question of AF, or at least this atrial remodeling or atrial myopathy, as said by Andreas, as a sort of expression of a more systemic disease, and also raising the question of which innate immune response is stimulated. Maybe nitric peptide have something to do with this story. So then so this is a sort of general scheme that have been proposed by the editorial of the journal, that there is a sort of, so they call that AF befits AF, which means that you have the sorts of accumulation of fat with aging, with probably lifestyle, things like that. And this accumulation of fat expose the myocardium to some immune or other process, and the group of Oxford also nicely show that this fat, there is a crosstalk with the oxidative status of the myocardium, and so slowly you can constitute a sort of fibrosis which also aggravates this sort of histological vicious cycle. So then the question is where this adipose tissue come from? What is the cellular origin? And that also tells a lot on this crosstalk between the two tissue and on the pathological significance. So when we look again carefully to this histology in human cells, we found that, so this is the epicardial layer and the subepicardial area. And as expected, this epicardial layer appeared a crucial area because there's a lot of stem cells, resident stem cells there, that you can stain with c-KIT or WT1 gene expression. Interestingly, some of these stem cell markers were also observed in the subepicardium, together with other markers indicating that some of these stem cells, epicardial cells, may also have acquired mesenchymal phenotype. And so some sort of transition from epicardial to mesenchymal cells can occur in this human atrial myocardium. And also interestingly, some of these cells expressing epicardial precursor markers were also detected in the proximity, in the vicinity of the adipose tissue. So the question was whether these stem cells in the epicardium can be a source of these adipocytes. So one way to answer this question was first to harvest these epicardial cells. There is some experimental procedure with this human atrial myocardium that you can harvest these cells. And in vitro, we have been able, of course, to induce a mesenchymal transformation of these cells. They acquire mesenchymal phenotype. And they also have pluripotent capacity. They can differentiate in chondrocyte, in osteocyte. But our interest is to see whether these cells can transform in adipocyte. And it's the case. It's very easy to induce adipogenesis of these epicardial derived cells by some control medium. And you see that there is this accumulation of lipids droplets, and there is a shift from pre-adipocyte to mature adipocyte, which is indicated by PREF1 perilipin shift. Then we go in mice, and we use a mice where we can follow these epicardial progenitor cells, where this gene WT1 is associated with a tomato dye, which give red cells. And so when we induce obesity in the mice, and now we found other circumstances in the mice where you can induce fat in the myocardium, you can follow the accumulation of the fat and see whether some of these adipocytes come from the transformation of epicardial progenitor if they become red, which is the case where you can see that some of these perilipin positive were also red cells, also well seen here, or some adipocytes become red, telling that they come from some sort of differentiation of epicardial cells. And again, when we harvest these cells in vitro, we were able to induce this differentiation in adipocyte with red adipocyte telling clearly that they come from some sort of epicardial differentiation. So then the question is, what are the factors that can trigger this differentiation and recruitment of epicardial stem cells? So we compare the screening of the proteins secreted by the epicardial adipose tissue or the myocardium on the same preparations. And it comes up in this screening that there is this natriuretic factors, which is, of course, as we know, highly expressed in by the myocardium. And you know that this natriuretic peptide is physiologically secreted by the EHRA in response to some sort of walking, loading conditions, and it's some sort of very physiological, relevant, mechanical biomarkers of the EHRA. So we try to address whether this natriuretic peptide can be involved in the recruitment of EPDC cells. First, we co-culture erythromyocyte isolated from the erythral appendage and human EPDC from the same patient on using the transwell. So what is secreted by the myocyte will impact on the EPDC. And indeed, we found that after a few days in culture, erythromyocyte can induce the adipocyte transformation. We also test directly a natriuretic peptide on these EPDC cells, and we found that for low concentration of the peptide, you have a very strong adipogenic effect. While for higher concentration of the peptide, you have a lipolytic effect, which results in the release of fatty acid in the supernatant of the solutions. And finally, we look at the expression of this natriuretic peptide receptor. There is 3 types, and we look at the which is the most expressed, and we found that these NPR receptors is indeed okay, so time is over. So just to finish with this scheme, this is a zero scheme where we are actually, that this fat accumulation or fat infiltration probably comes from some stimulation of the epicardial cells, but there is maybe other source. This is a sort of physiological process with aging, lifestyle, etc. But the fact that you get this accumulation of adipose tissue can expose to some transformation of the neighboring myocardium through, for instance, fibrosis, but there is probably other crosstalk between these two tissues, which can be stimulated by cytokine, adipokine secreted by the two tissue, and explaining that you have a sort of slow process of atrial remodeling associated with fat and maybe mainly in obese patients. Thank you for your attention.